All right, so we're going to continue talking about TDA with time series analysis. And let me actually give the most simple example before we come back to the memory reference trace. So here is a general pipeline, and let's start with the most simple case of one-dimensional signal. Let's say my one-dimensional signal is a curve that is coming from sine or cosine. All right. And then the first question is, how do I apply topological tools to study this kind of periodic signal? Okay. So the first question is to how to build a filtration. For persistent homology, okay? So of course, in practice, what you typically have is instead of having this very smooth curve, with infinite number of points, what you really have in practice is usually a sample from this curve. So let's say for simplicity, I'm going to sample this signal sort of in a uniform interval. So this now became my, but actually signal, which is a bunch of points where each point has an x and y coordinate, where x is in some sense, another way to write it is x is its sort of parameter and y is a function of this parameter, okay? But how do I map this to, say, a d-dimensional point? And let's say my dimension for the time being, let's say the dimension is 5. Of course, this is a dimension which actually in my notation is also what I call the window size. And then this is what's called Tarkin's embedding, or sometimes it's also called delayed window embedding, right? So the whole idea of having a window is if you have this sort of sequence of values, I would like to take a window of say size 5 and then take every 5 point and map that into a signal. Okay, so let's say at location 1, 2, 3, 4, 5, it has function value, right? This is for lack of, let's say this is 1, 3.5, 5, 6. And then again, 3.5, this is 3.5 and so on. So when I take the window of size 5, what do I get? I take the signal strength, right? In this case, f of x. So I have 1, 3.5, 5, 6, 3.5. So that became my first point in my 5-dimensional point cloud. And then the next thing I do is I also provide, let me use G as sort of the gap parameter. So this is window size, W is window size, and G is gap. And the idea is how long do I shift, between windows to obtain an embedding, for example. So for example, in this case, let's say it's equal to 1. So I basically shifted my window size by 1. So I start with 2, 3, 4, 5, and I include the value of 6. So then my points will be 3.5, 5, 6, 3.5, and then let's say this is like 1 again. Well, of course, this is not that interesting, but in practice, you may have more, varying values. Anyway, so the idea is as you go through this process, you are going to map a one-dimensional signal to a 5-dimensional point cloud, okay? And in this case, you can actually write a very simple code, even use using Python to actually just visualize and project this 5-dimensional point using things like PCA. And depends on the window size you choose. If you choose appropriate window size and you project it, what you will get when you project this 5-dimensional point cloud is actually you're going to see a circular structure. Okay, so that's where, so this point cloud, which is in 5-dimensional space, is actually where we can apply topological tools, right? In this particular case, I don't necessarily even need persistent homology. I might only need homology to say that, look, you know, there is a nice periodic behavior, and in the high dimensional points, it's actually a loopy structure, okay? So the periodicity directly translates to loops in your data. But the interesting part of using topological tool, either homology or persistent homology, is the actual computation of this or detection of this loopy structure or tunnels is actually can happen in the original high dimensional space. What I'm saying is that, of course, in this high dimensional space, you can project it and you can sometimes see it, but it's actually not guaranteed. It depends on the projection direction and depends on, you know, how regular that periodicity is. But actually what you can do is you can say, okay, I'm just going to apply homology or persistent homology in the original space. And then later on, we're going to talk about the specific concept of cohomology, which is the idea of assigning a parametrization to this point for specifically visualization purposes. But the idea is I can use sort of topology to detect specific the persistent homology and homology to detect this loop in high dimensional space, and this loop corresponding to periodicity. And what you're going to see later is actually we detect this loop in the original high dimensional space, obtain what's called a parametrization, which is, for example, if I call this high dimensional point cloud to be X, and then we say that there is a particular loopy structure, what we want to do is we want to apply what's called parametrization, which is taking a function and map this point cloud, right? Remember, this is a point cloud. And for every single point, I would like to map it to S1. So what is S1? S1 is a topological space that denotes a circle. So when you map this as a parameterization, what do I see is to say maybe starting from this point, this has a parameterized value of 0.1. You know, somewhere here is 0.2, somewhere here is 0.3, and so on and so forth. And then this thing here maybe is 0.9, right? This is what's called a parameterization of, you know, meaning that for every single point, I'm assigning, another way to assigning it is to assign a value between 0, 1, where 0, 1, the 0 is identified with 1 to create a loop. Okay, so idea is this parameterization tells me how the points are in fact distributed along this sort of loopy structure. Okay, so any questions so far? Okay, so now what do you see? If you look at going back to this picture, when I say there's a parameterization, this is actually an example of the visualization of the parameterization. So the underneath point cloud, you know, start with it doesn't have color, but then you use, say, homology or persistent homology to say, hey, there's a loopy structure. Then you can use a tool, like I said, called cohomology to assign a parameterization. So what you're showing here is literally the visualization of this parameterization using the rainbow color map. Okay, so perhaps the starting point is right here, where all the points goes around the circle is parameterized between a value of 0, 1. Okay, so, and of course, once you have this parameterization, you can map the 0, 1 interval into what's called rainbow color map. So if you follow the rainbow color, that gives you essentially ordering of the underlying points to see how the loop structure are formed among the points. Okay? But of course, there is this question is, okay, so where we're alluding towards too, which is actually not covered today, but in a later lecture, is that this parameterization is in fact a form of visualization for dimensionality reduction. Okay, so of course you can argue that if this loop looks this relatively nice. I don't really need the sort of parameterization, which in this case, the coloring, to tell me that's how the loops looks like, because human perception can tell me, oh, of course, when I projected this point cloud, I see a loop structure. But the most interesting part is actually this parameterization happen in high dimensional space. The detection of those loopy structure happen in the high dimensional space, which is in this tokens embedded space. And it's actually in some sense independent of how those points are projected. A very interesting example actually is that I would like to take a wire. Do I have a wire? Extra wire. Just a second. Here we go. Okay, let's talk about how the loop structure form in high dimensional space. Not all loopy structure in high dimensional space, of course, my high dimensional right now is 3-dimension, right? Not all loop is going to be in a perfect loop like this. And then when you project with PCA, you see a perfect loop in 2D. Very much so, this loop can have twists like this. I mean, it's still a loop, but it has an interesting twist, for example, okay? So now what's interesting about it is that depends on how the projection happen, the projection itself without this parameterization may create illusions. What do I mean by that? If I have, let's say I'm just giving you a very simple example of a 3-dimensional space, and then there's a loop in the middle. And then of course, if this loop is as perfect as what I was drawing early, that if this loop looks like this, then when I project it in a proper direction, you see on the screen a pretty good loopy structure. However, if I have a loop that looks like this in the high dimensional space, then the interesting question is, when you project, what do you get, right? So you can imagine there's some direction when you project, for example, this direction, maybe what you will get is this. So everything is good. But there's a lot of directions, actually, when you project this. What you're going to see is something actually looks like a figure 8. All right. So imagine you have a point cloud sample from this shape, which is a little bit complexity, a little bit curvature. You might get very different understanding of the point cloud by looking at the projection. In another way, that there might be projection direction you get that actually give you the illusion that my data actually contains 2 types of loops. However, if you actually parameterize this by a rainbow color map, for example, so if I parameterize again this point cloud onto S1, this is something, no matter how you project the data points, you are still going to see by just following the rainbow map how that loop is formed. All right. So what's going to happen is that even if you have a projection that is of the figure 8 shape, but then if you apply a rainbow map to it, the rainbow map will just go around this loop in a rainbow color. And that gives you the idea that, okay, even though I am seeing a figure 8, but following the rainbow map, it's actually only one single loop. It just happened to be while doing this projection that the loop kind of fold onto itself. Okay, making sense so far?
So, I have a question.
Yeah, go ahead.
So basically the parameterization is bringing it down to 2 dimensions?
No, the parameterization is a parameterization of the points in high dimensional space. So for every single, let's say this is actually indeed a 5-dimensional, well, actually what I drew is a 3-dimensional point cloud. So for every point X, Y, Z, if I give a coordinate on this curve, it gives some values between 0, 1.
I guess.
Yeah, this parameterization is in high dimensional space. The visualization if you have a way to visualize high dimensional, there's nothing prevents you, but visualization is to say, I'm projecting this point into low dimensional space, but I carry over this parameterization and visualize this parameterization in the projected space. So
I get it. I just don't get the, like the picture on the previous slide
yeah.
that has things that are off of the actual circle. Like I don't get what that dimension is.
That's actually interesting, right? So when you think about a point cloud, actually this is something, this is a specific cohomology lecture that I'm going to give. So what's happened is that my point cloud is not necessarily going to be perfect like this. This is actually a point cloud sample from some high dimensional thing where there's actually a lot of noise surrounding it. So another way to think about it is that my point cloud in this example is actually annuous. Okay. But for me, when you think about a homology, what is homology? When compute homology, it's saying that, okay, somewhere here, there's a loopy structure. But if I want to describe the loopy structure involving the entire point cloud, what do? The loop structure still goes around like this, right? But what I would do in this parameterization is instead of just assign a parameterization to a subset of point, I'm assigning the parameterization to the entire point cloud, such that, for example, in all this area, like I drew here, all those points in this area, it's roughly a particular location with respect to the loopy structure, and then they are assigned sort of values of similar ranges. Does that make sense?
Yeah.
So X is not just a subset, X is the entire point cloud.
Gotcha. I get, and so the last question is, when you say, there's noise, it's not perfect, there's noise, but what dimension is there noise? Or is it noise?
It's in high dimensional space.
Okay.
Everything, so other than the actual projection of the point cloud, everything else like homology computation, persistent homology computation, parametrization, all of those happened with respect to the high dimensional point cloud. It's only that when want to visualize this loopy structure in high dimensional point cloud, I'm turning my parameterization into, say, choose a particular color map and just project the point together with their color.
Okay.
Does that make sense?
It does. Yeah.
Yeah. Okay. So actually, just to give you a better sense of this, there's actually, I kind of reserve that for later lecture on cohomology. But since you're asking, let me just show a few pictures of how this looks like. So let me just share a different screen. So this should look very familiar to you. This is a point cloud coming from your first mini project. This is in fact a sort of, you can think about all the points from the surface of the mesh to form a three-dimensional point cloud. Okay, so this is what I call the octa data set. And then what it really is actually the shape of a bow tie, well, with four large loops, but actually there's also minor loops in between. But what's interesting about it is that when this is a three-dimensional point cloud, I can use, say, tools from homology and persistent homology and cohomology to try to detect 4 different loops. And for each of those loops, I'm going to try to apply a parameterization of the entire point cloud, but it's emphasizing on each individual loops. For example, if I want to emphasize this loop on the on the upper right corner, the rainbow color goes around this loop. But because I'm like I said, my parameterization is only focusing on a particular loopy structure, the rest of this has the same color, which means it's something I'm going to ignore, right? So pretty much my parameterization really goes around this, and everything that is surrounding this green point is all green. So my visualization really highlight this loopy structure surrounding the rainbow map. I can do the same thing for each of those individual loopy structures. And this is actually what you see when I project it into two-dimensional space.
Okay, this makes a lot more sense now.
Yeah.
Okay.
So now there's actually another picture, which this is an interesting thing. This is where persistent homology comes in, okay? So again, this is an example I was showing you before. I have a point cloud, and imagine this point cloud is, for example, a point cloud sample with quite a bit of noise. Then when I'm detecting the loopy structure, I have different persistence associated with those loopy structure. So the left one, of course, is a one with high persistence, is a large loop, and I have a rainbow color surrounding it. But of course, because I can use persistent homology, which means I can detect the loop structure at different scale, right? So in this case, I can also detect, say, a low persistence loop. And remember, my parametrization is a rainbow color. So I will apply rainbow color surrounding this, but then for the rest of it is one uniform color because my rainbow or my parametrization only focus on this small tiny loop. So you see that when you project it, you see that, well, maybe on the left is my high persistent loop and its parametrization, on the right is my low persistent loop and its parametrization. And the rainbow color kind of highlight where that loop is. Okay. And I want to see, yeah. And then this is actually what's interesting. This is actually the memory reference trace projection without me kind of try to map them to actually to be a loop. Later on, if you see this is a kind of visualization we created based on this parameterization, just to emphasizing there's loopy structure. But in practice, this is actually what you see, which is actually quite interesting. In practice, this is sort of the reference trace. This is on the left is a small trace. Again, I'm taking the high dimensional point cloud and project it and try to kind of get the idea. Overall, I mean, this trace isn't that long, so I actually didn't complete this loop. But what's interesting is that overall, if you look at the shape of it, there is roughly a loopy structure. But then at every single location, there's a tiny little loop going there. And this is an indication of multiple periodicity from my signal, right? There's a large periodicity, which is a large loop. And then there's a different frequency at which corresponding to this little loop. And if I actually project it using, so the left is, this is one trace I project. And then on the right side is another trace I'm projecting. Again, I have a periodicity mapping, and you see this sort of periodicity coming from actual projection. So in some sense that this parameterization is now independent from how I'm going to embed it, even though the embedding isn't perfect. But if you look around, follow the rainbow color, you see this is actually a loop in high dimensional space. All right. So I don't know if I have the picture in here, but one of the classic image, I think it will come back when I'm talking about cohomology, but let me share. So let's have a quick sort of mental exercise, right? What's going to happen, actually, I might have this picture. What if my point cloud is sampled from a torus? Okay, it's a three-dimensional point cloud sampled from torus. We all know that, for a torus, there's two important loopy structure, right? There is a big tunnel, and then there is a tunnel that goes around the cylinder part of the torus. So one interesting thing about it is no matter how you project the torus point cloud onto two-dimensional plane, no matter how you do it, you are only going to see one of those loops, right? Does that make sense? Because the loop that is surrounding the cylinder, it's very hard to see, okay? So that is really the interesting idea of how you, yeah, so this is a good picture. Let me share with you. So this example from the original cohomology parameterization work, okay? if you compute the persistent homology of a point cloud coming from a torus, you see that there are 2 points in dimension 1. Those corresponding to the 2 loops of the torus, right? One going around this tunnel, or one goes sort of as goes around the the cylinder. So when I parameterize it, actually this is what's interesting. So on the right hand side, this is a classic parameterization that goes around the middle tunnel of the torus, while this picture on the left is a rainbow color that goes from interior to the outside and goes around. So this is trying to, I mean, not perfectly, but it's trying to indicate the other loopy structure goes around the cylinder part of the torus. Does that make sense? Yeah, so that's what's interesting about this is that, you know, can people see my slides? Oh yeah. So basically, if you have a torus, you know that there is my red tunnel, and then there is my green tunnel. And then the picture I just showed you is trying to use user parameterization, which is corresponding. So what's happening is that there is going to be 2 parameterization. There is a first parameterization that parameterize. Well, there's a second parameterization the entire point into S1 for the blue circle, and then there is a parameterization of the entire point cloud that parameterize the point cloud with respect to the first circle. So as you see in the visualization, for theta 1, you see the rainbow color goes around the main tunnel. For theta 2, you see the rainbow color goes around the green tunnel. Okay. Anyway, we are a little bit ahead of schedule in terms of explaining. I want to actually give the mathematical notation of this, which is called cohomology, which is in some sense, the dual of homology. So one thing that I think mathematician likes to do, especially in algebraic topology, is that you have homology, you have cohomology, and you have chain, you have co-chain, you have boundary, you have co-boundary. So pretty much the idea of a cohomology is a dual version of homology, but with the added benefit, it's actually instead of like the chains are in homology, it's linear combination of simplices. The co-chains are maps on those linear combinations. So there is a very complicated, well, not say complicated, there's a very intricate mathematical formulation. So that by itself deserves a single lecture to discuss about. But for the time being, let's say what we get here in terms of parameterization of mapping a point cloud to S1, this is a result related to cohomology computation. All right, so now let's talk, coming back to our memory reference trace. I said, okay, I can have this one-dimensional signal and I can map the, like, you know, every say 5 data point to a 5-dimensional point, and I shift this window. Then depends on how many window you have, you get the size of your 5-dimensional point cloud. In the memory reference trace, my signal is those memory address. Okay, so I take, in this example, I say I'm going to take every 4 memory address to form a 4-dimensional point cloud. The important part, of course, in this example here, if I want to kind of map it to here, I can use, for example, Euclidean distances between my point cloud, points in my point cloud. But here, because they are memory reference traces, you can use things like edit distance, for example, which is how you modify from one string to another. So essentially, every single actual address is mapped to an alphabet. And if it's the same address, it's the same alphabet. And if you take the 2 points in this point cloud, their distance is edit distance. So this is what we mentioned at the end of last lecture, is, for example, the distance between the string, this 2 string is equal to 1, because it just take 1 modification. Usually the edit distance have the modification include deletion, insertion, and modification, for example. But you do have some flexibility in defining how you design as edits between strings. There's also a similar, so for this, our purpose, actually using edit distance, achieve what we want. So that's where we go with. So now this is how you get what is a visualization, right? So what we see is that, if I do see a circular structure, I'm also, in addition, so this is slightly different from just visualizing the point cloud as a perfect circle, because I want to also capture the time. So basically, for every single point goes around, I'm basically visualizing. So now every single point in this visualization corresponding to a high dimensional point from this tokens embedding. But I also kind of involve them along the time axis going from center to the outside. So this, going from center to the outside, this is what I call the time axis. So using cohomology, I obtained a parametrization of this point cloud into S1, but then I append every single point with its time axis. So instead of having them just go around a perfect circle, I pretty much want to increase the radius to capture the time, okay? All right, of course, going around this direction is what I call parametrization. This is precisely what we obtained from studying this loop structure in high dimension. Okay, and then what I'm treating each of the node, like I said, this is a high dimensional point coming from the memory reference trace, and I'm calling it a memory event. Okay, so now what do we see, right? I mean, I kind of feel like I did this project many years ago, but I feel like this is a really cool and sort of non-conventional application of topological data analysis. And it's actually, I mean, we have a lot of interesting directions where we want to go, but we haven't really pursued it since then. But the idea is following. If you see this example, this is a bubble sort, okay? And the first question we ask ourselves, if it's the same exact program of bubble sort, but my data is either sorted, inversely sorted, or random, do I see a difference in my memory reference trace using topological techniques? So in this particular case, this is a first example where the data is already sorted. So if you look at the algorithm of bubble sort, the only thing you are doing is comparison. There's no swap because it's already sorted. So now if you look at the primary, the dominant sort of highest persistence loop in this high dimensional space and map it to my visualization, you see there's 4 loops roughly, which corresponding to those 4 things goes around 4 times, right? So 4 loops, sorry, I want to say there's a little bit this loop means that there's 4 event in this 4 loop. Okay, so and each time I go through this loop, I go through what? I go through 1 comparison. So there's 4 comparison, which then corresponding to, in my actual visualization, those 4 loopy structure in my visualization. Okay, so now the next thing is, what if I have an inversely sorted structure? Okay, my inversely sorted structure is each time I do a comparison, I also have to do a swap. So now each of those red box corresponding to both a comparison and a swap, right? Because 5 is bigger than 4, so I need to swap 4 and 5. And then 5 is bigger than 3, I need to swap 3 and 5. 5 is again bigger than 2, swap, 5 is bigger than 1, swap. But this is because my data is inversely sorted. So pretty much this is, in some sense, is a worst case scenario for my bubble sort, because I need to pretty much go through all pairs. And what you see here is that your comparison is still those loopy structures, but then the little teeth, right? The teeth here, I mean, you see this very thick teeth, it means there's a lot of memory events in that area that correspond to the swap option. So the number of teeth corresponding to how many comparisons you are going to see. Okay, so if you go, remember you have to go from the center because that's the beginning of time. You see that there is 4 teeth, which is a 4 swap. There's 3 teeth, which is a second range of 3 swap. And then there's 2 teeth, there is 2 swap, and then there's one teeth, which is one swap. Okay, so this is very interesting that even in one single parameterization and visualization, I'm going to capture both comparison and swap. And of course, naturally, the next thing is, what if my data is sort of permuted? Okay, it's not perfectly sorted. It's also not perfectly inversely sorted. So it's actually kind of a random set of number. So there is a number of comparisons, and then there's a number of swaps. So again, it's the same thing. In this particular visualization now, is that the loop is captured both like comparison and swaps, and the teeth is just the number of comparisons. Okay, so for example, in here, I have going from inside to outside. I see the first segment, I don't have any swap, but the second, any comparison. So remember, the blue is comparison, red is comparison and swap. So in this case, the blue, I have two comparison that corresponding to the two things here. Okay, so what if I put them together? If I put them together, those are in some sense my sort of topological structure I captured using my parameterization, my cohomology parameterization, and then this is sort of the different visualizations surrounded to different input data to the same algorithm. Okay, and now let's move on to the next one. There's also what we want to investigate is algorithm-dependent structure. So what is algorithm-dependent structure? Let's say we take the example of matrix multiplication. So matrix multiplication, this is sort of the naive matrix multiplication, okay? So basically, you know, I mean, hopefully everybody recall what is the matrix multiplication. When you actually multiply two matrices, right? You need to take a row of the first matrix, column of things, you multiply this with the first one, the first element from the row to the second element from the column, sum them all together, right? So this is roughly sort of one version of implementing the matrix multiplication. Essentially, it's 3 loops, okay? And you kind of add them together. So what's interesting about it is that this is one version of the matrix multiplication implementation. And then this loopy structure you see is actually the parameterization, the 3 different parameterization of the same point cloud. So this is essentially thinking about the torus example. I have theta 1, theta 2. This is essentially correspond to, this is theta 1 going from point to S1. This is theta 2 going from point to S2. This is theta 3 of point, to S1. Okay, this is a 3 different parameterization of the same point cloud. And then they correspond to 3 of the highest persistent points in my point cloud. So what do I have? I do my persistent homology magic. I get a lot of noise, but you get 3 points. So you can basically separate signal from noise by looking at those top 3. This is the top 3. This is the top. So remember, each of those top 3 points corresponding to a high persistent loopy structure in my point cloud. Each one of them give rise to a parameterization. And this is a 3 parameterization visualized in our special tool. And what's interesting about it also, this 3 visualization corresponding to the 3 nested for loop. Okay. And then in addition, you also see this kind of almost banded operation here. And this sort of captured in the 3rd loop how those matrices are multiplied. Okay. So hopefully, because I'm talking about matrix multiplication, you know there's an obvious optimization of matrix multiplication, which is called block-based matrix multiplication. Okay. So the difference between this one and this one, this is a block-based matrix multiplication. So it's a small optimization of the class, the naive implementation, which is when I am multiplying 2 matrices, I'm going to sort of divide the matrices into blocks and do the multiplication on a block base. And that has some sort of certain memory pattern that is different and also is actually more optimized. And what's interesting about it, again, I'm going to pick the top 3 highest persistent loops in this high dimensional point cloud, parameterize it and visualize it. And this is what you see. Again, this 3 structure corresponding to the 3-4 loop. But the most important part is you now see a very interesting sort of, bundled pattern for the last bit of this multiplication here. So if you put them side by side, the top is a naive matrix multiplication algorithm and its memory reference trace pattern. And then the bottom is a block matrix multiplication. And you can see a very different kind of signal to it. Okay. So this is sort of like one of the things that we have kind of spent time looking into. And actually one of the things I thought it would be interesting, sort of almost like a machine learning / exploratory analysis, is to say that, if I can use topology to capture those patterns, can I use those patterns as some sort of diagnostic tool, right? Can I use this pattern to inspire more like sort of code optimization? Okay, I mean, that's sort of our crazy idea. And our funding agency thought that we're a little bit too crazy, but I still think it's a very valid research direction. Okay, so let's look at the last example. Oh, actually, I didn't show it. I want to show it here. Let me show it here. I didn't put it in the slides, but I still think it's interesting example. Yeah, so this is one more example, and then we are going to take a quick break. We even went into a more extreme example of looking at MPM, which is sort of a multiple, it's a sort of a particle tracing algorithms. So you have multiple particles in your code, and on this again, you run this piece of code, right? In this case, is you are going to look at sort of you're looking at the execution of this code, and now what you are looking at is, if I look at this code, of course there is, well, obvious periodicity behavior is always coming from for loops, but there's also things that is related to like how many times. Oh yeah, here, sorry, this should be the code. So this is MPF code, and then there is again another type of periodicity where it's not just coming from sort of just a for loop, but coming from essentially calling the same functions. Because what you could imagine, if I'm calling a specific function sort of multiple times, you can also see a sort of periodicity behavior. And so you can actually see, again, from this process itself, you can see, again, the periodicity that is coming from repeatedly calling the same subroutine. Okay. And finally, I want to mention that Finally, I want to mention that one thing that you have to deal with for this particular project is memory reference trace is long, right? If you execute a tiny program, you get a really long memory reference trace. So of course you are dealing with scalability issue because you're taking the memory reference trace mapped to a high dimensional point cloud, which might be huge, and then you are going to apply things like persistent homology to it, right? So how do I scale it up? Because actually the best, if you run Ripster, you will know if I have, I don't know, like even 1,000 point, your number of simplices will reach to the million. Okay, so very small, like in some sense that the size of your simplices grows quite quickly, and which means that you need to have a very scalable approach. So one type of scalable approach is basically in the memory reference case is actually quite straightforward, is to subsample, right? So you want to subsample the memory reference trace so that it's kind of became a point cloud size that is reasonable to be handled by persistent homology. However, so what we actually kind of suspected this framework would be most useful is not to say, okay, give me a very complicated program, taking the entire memory reference trace and try to see the periodicity, because you know those periodicity is actually localized to certain part of your program, right? So one thing you could do is using this as a tool to kind of study maybe a particular snippet of your large code, right? You say that, okay, you know, maybe there's a piece of code I want to see its behavior. For example, the piece of code I'm looking at in the slides is a matrix multiplication. So I only want to look at that particular piece of code and to see what the memory reference behavior is, right? I mean, that's also aligned well with people who study like behavior of program. You don't just take the entire program, taking the entire memory reference trace and try to understand the global behavior, but much rather you are always sort of performing more of a diagnostic kind of task to say, okay, if there's a small snippet of my code, I want to understand how it's behaved. And then I want to say even a little bit more where this research can go is that we are looking at memory reference trace, right? I mean, you can capture something that is behavior of software that goes beyond just memory. I mean, memory is sort of the lowest level access pattern. You can also think about access pattern that is related to, say, your cache, right? What is the read and write of your cache, and so on and so forth. So in some sense that this framework, you can kind of apply it to sort of different level of the system and collect the signal from different level and try to see what you can detect. And then finally, again, I'm going a little bit ahead of time, but let me share with you one more thing. The tools that we use here, you're like, okay, you know, I map it to a point cloud. I use persistent homology to detect the sort of loopy structure. But, you know, like a memory reference trace isn't just loopy structure, right? What about a conditional statement? If then else, what did that correspond to, right? I mean, again, you know, homology, if spatial dimension one homology is detecting loops, but there has to be something else that is structure-wise that is interesting to detect. You can imagine there can be tools, again, topological tools to detect other kind of patterns, for example, branching, right? If then else is essentially a branching structure, right? So if you think about, well, literally, if I try to capture that, and visualize it, I will look at different branching structure. So again, just as a sort of small advertisement for our lecture on cohomology, we're also going to talk about using a similar framework to study the branching. And it's going to be, again, let me share this. So I just want to show you one example. So this example here is using the same framework of cohomology, but the idea we focus is not the loopy structure any longer, but the branching structure. So as you see, again, this is a parametrization, okay? The parametrization says the rainbow color goes around each of my branch. So, but of course, based on the relation between cohomology, when you actually have what you imagine a three-way branch, it's actually really branching twice, right? So you actually have like two directions where it's branching. So this is what the color is capturing. So you see this rainbow color saying focus on the red. This is one direction it's branching. This is another direction. Going back to our point cloud from the first project, you see that in the middle, right, this bow tie, I actually have a 7-way branching. So each of the parameterization is tell you which direction is branching to. So again, this is using cohomology, and you're roughly using the same idea. And you can see from this teaser image what it does. If I want to detect two-way branching, I look at the local neighborhood of the tip of my point cloud where I want to detect whether there's branching from that point. I take a local neighborhood. In this case, I take a high dimensional ball and just looking at all the points within a distance from that point, right? So that's all this color point is. Now I want to detect the branching, but I can translate the branching back into detecting homology, which is loops. what you do is you have a three, like this is two-way branching. I get all these points that is in this neighborhood. What I do is I treat all the points on the boundary to be glued to a single point. Okay, so if I go from here to here, when they all glue to a single point, you see the picture B, right? It's all this points cloud, and I kind of identify the boundary points to be a single point in the algorithm itself. Then what you see, in order for me to detect branch, I can detect how many tunnels do I have. In this case, I have two independent tunnel, which corresponding to two different branches. Okay, so going back to memory reference trace, what did this imply? Which actually is something we haven't done. If we take the memory reference trace, what if my goal isn't detecting the periodicity now? My goal is to detect sort of bifurcations in the program. This will be the corresponding topological tool to detect that. Okay. All right, so I'm going to take a quick break. We'll come back in 5 minutes. Any questions during the break?
There's a question in the chat about block matrix multiplication. So does the block matrix multiplication...
And then when we come back, we are going to talk about a bit detail over...
Hi, Bei, can you hear me?
Over mice data.
Hi, Bei, can you hear me? Sorry, I think I was... Hello?
Can you hear Faye? She's talking.
Hi, Bei, can you hear me? Hello?
Yeah, we can hear you, Faye.
Oh, okay. So there was just a question asking if the block matrix multiplication requires some special parameters or characteristics. But I think that's a linear algebra question. I don't think Bei can hear me right now.
Nope.
I was trying very hard to talk, but anyway. Yeah, but I linked.
Wave your arms frantically at her.
Hi. She's not paying attention to me. Can you hear me?
Are you trying to talk to me? I couldn't hear you.
Yes, I've been trying to talk to you in the past like 3 minutes.
What's that?
Yeah, there's a question in the chat about the block matrix multiplication. So does the program that you introduced, can any type, like any block matrix multiplication apply like to the framework, or does it require some special?
This is a tool, right? This is a tool for you to study memory reference trace. So any program or any sub part of your program, you can collect the memory reference trace. The matrix block multiplication, right? Maybe there's other implementations, then you can actually play with their memory reference trace, apply the exactly same framework. The slides I was trying to demonstrate is to say, look, if I actually change how this particular algorithm is implemented, one is naive and one is slightly more optimized, we will observe from the actual visualization the difference between them. That's what I mean. Does that answer the question? Yeah.
Yes.
Faye, for some reason I didn't hear you. Like for a while, I only happened to see your motion, but I didn't hear you.
That's fine.
Sorry. Yeah.
That's why I started waving.
I know. I mean, that's why I noticed. I was like, why is Faye waving? I couldn't hear her. Okay. All right, let's get started again. For some reason, my video is okay. So, okay, so I want to give a different example, and then this is sort of a classic time series data. And in this particular data, we are talking about the core body temperature of mice in the lab. And then my collaborator who studied this kind of data is interested in sort of study of sort of biological clock, right? So you can imagine that in this particular case, what I'm showing is a segment of the sort of the body temperature measurement for a mouse subject sort of before, during, and after pregnancy. So in this case that you see on the upper left corner, this is sort of the temperature data, like the original raw temperature data. And again, remember I said there's some sort of scalability issue if I have too long a signal, if I want to apply persistent homology tool, I want to maybe reduce the size while preserving the signal. So you can subsample and you get the picture on the upper right corner. And then that still capture roughly the behavior of the temperature data. And then here, there's also a bit of, what's the difference between these two? Okay, sorry, just a second. Let me show you. Oh yeah, so first of all, okay, so the difference is that, in the original raw data, we can have some potential outliers, right? And those are sort of outliers. So your first thing you need to, there's a lot of sort of pre-processing to deal with this kind of data. And here is sort of two mouse subjects. And this is sort of after you do an initial cleaning, you get the middle picture. And then you need to also do a little bit smoothing. You get the third picture. And then the fourth picture is a sampling. And then what's interesting about it is that we actually tried both framework for us to get, sort of nice signature. What we actually did, which is a little bit aggressive actually, and we put a lot of discussions over this, is I can also apply additional what's called rank filtering. So rank filtering is a tool that, for example, in this particular case, we kind of preserve the dominant frequency of my signal, right? There's a lot of filters that is actually used in signal processing. And it's interesting to actually even experiment TDA with different signal processing tools. And you can change different filters. In this particular case, we preserve the dominant frequency of the signal. And then we apply the persistent homology computation to look at the most dominant or high persistent circle structure and then map it back. So what you see here is a visualization of, again, my tokens embedding. And then the color, the rainbow color, correspond to the periodicity, okay? So going back to here, what you see is actually, right, the question is, well, if you were going to take a guess, this is the high dimensional point cloud of the mice temperature data, and then parameterizing high dimension, well, I mean, it's pre-processed, including using rank filter, and then project it into low dimensional space, okay? If I were going to ask you to make an educated guess, who do you think is, well, there's actually 4 subjects in this picture, right? So which ones do you think are the ones who get pregnant? Which ones are the ones who are not pregnant? If you were going to take an educated guess.
Chat says right side pregnant.
Yeah, exactly, right? So why is this the case, right? So in some sense, pregnancy actually changes a lot of hormone behavior in the mice. So it also sort of in some sense interrupt its regular biological clock. So that if you measure it by temperature, you can actually see, and in fact, actually the measuring of body temperature change is one way actually for a pregnancy test, is that there is very detectable changes. Okay, so that's really what you see. I mean, of course, you can also see there's a little bit variations among them. Now, this is interesting. Okay, so not only in the experiment, they are looking at sort of mouse that are pregnant versus not pregnant. They're also looking at mouse who are jet lagged and not jet lagged. Okay, so it turns out when a mouse is subject to jet lag, it also changes the sort of its temperature behavior. And this is some examples. So to be fair, this is actually, again, a loopy structure is just we're projecting using principal component. So this is PC1, this is PC2. So this is projected over the first two dominant direction of the principal component. But if you project it of PC2 versus PC3, you're going to see the loopy structure. Again, the color, the rainbow color goes around this dominant loopy structure. Okay, so not only, so the interesting experiment that biologists has collected is not just pregnant versus not pregnant, jet lag, not jet lag, but also subject pregnant mice to jet lag as well. So to complicate the situation. So I would like to show you a bit more detail over some of those. So this is something we recently submitted, which is a collaboration with Ben Smar at UCSD. So first of all, let me show you the raw data set. There is 104 mouse subjects, and then each column is sort of the temperature collected. And you can see the raw data, and then this is true for a lot of sort of practical applications. The raw data has discontinuities, outliers, and so on and so forth. So it has to be some initial process of processing this data. For example, for some of those mice, if they have a really large gap here, when we are constructing the high dimensional point cloud, we actually treat them as individual sequences and generate point cloud for each one of them and then concatenate the point cloud, right? Because you don't want sort of the tail end and the beginning to be affected by this. And of course, for small gaps, because this is actually quite long, in fact, some of those data set contains essentially, sorry, let me give you the statistics. It contains more than 10,000 points, I believe. So you actually have a quite a long signal. So yeah, so the largest one, some of them is roughly 95K data points. Okay, so that's why we also need cleaning. And so if we look at this data, what you can do, this is something we haven't talked about, is that you can take the point embedded in high dimensional space, parameterize it. You can also bring the parameterization back to the one-dimensional signal, okay? So this is sort of a highlighted version. Let's say I have the parameterization of this. So every single point in my high dimensional point cloud now is mapped into S1, right? And what I can do is I can bring the parameterization back into my sort of, this is the data that is after some form of rank filtering. So now you can see the rainbow color goes up and down. It's quite dense, but basically the rainbow color goes up and down like this to really capture the periodicity of the signal, okay? So that's something that people commonly do when they study TDA and signal analysis, is you can also map the loopy structure back onto the original data set, it's original time sequence, okay? Now, actually what I think is really interesting, this is what we see versus left is non-pregnant subject and right is pregnant subjects. And this is all the mice that is jet lagged but non-pregnant. Okay, so you see a bit of a diversity over how their signal is influenced by this jet lag, right? This is why, this in some sense, if you imagine when you take a really long international flight while you're feeling jet lagged, I mean, this is really because your underlying biological clock was disturbed by the jet lag event. And then there is individual variability among those jet lag events. And what you can do as part of combination with this and data analysis is by using those topological profiles of the mice, you can also apply clustering. So the idea is I would like to study the subpopulation that have slightly different behaviors. So in this particular case, this is a very simple example. You can use, number one, you can cluster the time sequence by using things like wavelet. This is really just taking traditional signal processing approaches, taking the original signal using wavelets, transform, and then cluster the corresponding coefficients associated with the wavelets. On the right-hand side, this is using dimension 1 persistent homology of those signals, right? So you can then study, right, if I were going to cluster them based on topology versus if I cluster them based on classic signal processing techniques, what are their differences? Okay, so that's sort of additional thing you can do. The next thing you can do is actually study now how jet lag affect an individual mouse. So what you see here is there's two subjects, both non-pregnant, and then we chop their signal into 3 pieces. The signal before, because when we introduce jet lag, there is a particular starting point where you're introducing jet lag. So I think if I remember correctly, it's like shining lights at them, which is a little bit crucial or brutal actually. It's a reminder of interrogation techniques that you can never go to sleep. But in this particular case, you take the sequence, you take them into 3 components, and you get the first part is a time signal that is before jet lag. This is a time sequence during the jet lag, and then this is time sequence after the jet lag. And you see that one, again, using my persistent homology together with the parametrization, I see, of course, before the jet lag, it's non-pregnant, you have the perfect periodicity. During jet lag, you are influenced by jet lag, so you get perturbed signal. And then there is what's called sort of a lingering effect of the jet lag. Even after the jet lag has officially stopped, it takes a while for your sort of biological clock to come back to normal. And this is example of, again, variation of even, so this is where things became interesting. We see that if I have no jet lag as a factor, there is a clear separation between pregnancy and non-pregnancy. But in here, even you just introduce jet lag to non-pregnant mice, you see that jet lag itself is going to create some sort of visual pattern that is actually very close to the visual pattern you see with pregnancy as well. So in some sense, it became a little bit hard. If you think about classification, become a little bit harder to classify when jet lag is involved. And then finally, this is sort of, there's also what's called, among the mouse subject, there's also what's called a partial pregnancy. So for mice, for some of those subjects, they were initially pregnant, but their pregnancy did not come to term. So that's what's called partial pregnancy. And you can also see the difference between partial pregnancy, mice who are partially pregnant, okay? And finally is now you introduce jet lag on pregnant mice. You see some variations, but again, purely based on visual inspection will not give you all the best classification, but it's nevertheless interesting sort of profile you can look at. For example, on top, those are pregnant mice who are not jet lagged, and then the bottom are the pregnant mice who were jet lagged. Okay, so that's what we did with sort of introducing the rank filter. But of course, the natural comparison is if I don't have rank filter. So let's look at the ones with rank filter. So this is all the non-jet lagged, non-pregnant subject. Okay. So as you see those special. Oh, actually, let me be careful here. If I put them side by side, the top one is not jet lag, not pregnant. On the left-hand side is when we apply rank filter. So of course, rank filter kind of only preserves the dominant signal. And because of rank filter, I kind of reshaped my point cloud in a bit so that I actually get very perfect circular structure. The right-hand side is without rank filter, right? Taking the signal and map it. And you see that, what I show here is projection in principal component 1 and 2 versus principal component 2 and 3. The idea is, even though you see this loopy structure, but if I use another projection, you could intuitively imagine those are actually more like cylinder structure in high dimensional space. Okay, so you see this perfect cylinder when I'm using rank filter, and you sort of see a little bit more like deformed cylinder when you're not using rank filter. But overall, you still see this very perfect circular structure. This is non-jet lag, non-pregnant. And this is the non-jet lagged partial pregnant. Again, left is with rank filter, right is without rank filter. And what you see in this particular case is that actually, especially when I'm taking the entire sequence for analysis, for partial pregnancy, it's actually still dominated by the signal that is coming from non-pregnancy, right? But you still see a little bit, if I don't use rank filter, you still see this deformation of, so in some sense that, you still see the effect. And then the last one is non-pregnant jet lagged, okay? So on the left-hand side is not pregnant, and on the right-hand side is, sorry, no, this is non-jet lag pregnant. Sorry, this is non-jet lag, not pregnant. So on the left is with rank filter, on the right is without rank filter. So this is a situation where there's no jet lag at all. Actually, this entire thing is not jet lagged. So on the left-hand side, you know that the pregnant mice have this sort of almost like annulus projection in one direction, versus if I don't use rank filter, it's actually non-jet lag pregnant has this really interesting kind of shape, right? And then the whole reason when I'm looking at the parameterization is actually also quite interesting. The parameterization really try to pick out this particular tunnel right here. And then this shape is essentially formulated because you know that when the mouse get pregnant, that the periodicity actually changes. And this is an interesting intuitive understanding. How does the pregnancy, like hormonal changes the biological clock in a very intuitive way, okay? So now let's even just spend a little bit of time talking about jet lagged mice. So jet lag is now making things very complicated. Again, left-hand side is with rank filter, right-hand side is without rank filter. So again, you see that, rank filter, of course, make my data a little bit easier, but it did sort of reduce some sort of complexity of it. So this is jet lag non-pregnant. This is jet lag pregnant. And, oh, sorry, this is jet lag partial pregnant, and this is jet lag pregnant. So what's really interesting when you look at the sort of the embedding, the PC1, PC2, PC1 versus PC2 and PC2 versus PC3 projection, you see this very interesting sort of visualization of the corresponding signal. Okay. All right. So that's what I want to say about mice. And we actually have, I think we're going to post a paper on arXiv soon, and then the data is also public available. So if you are interested, you can play with this data set. For the last 5 minutes, I want to mention some other applications in using TDA for time sequence analysis or time series analysis. Well, in this case, again, it's not what you consider the really time series, but also like time varying data. In this particular case, we have a time varying social network. And then this is a simple social network where it's a social network of emails in a high school. Okay, so what you do is that at every single time of the week, and also time of the day, there's like you break out each day into say 24 hours. And for each hour, you collect sort of number of emails sent between A and B. And then that's how you construct the email network where the nodes are the faculty students in the high school. And then the edges is at where there's email between them during that hour. So you have a time varying social network. And the large picture idea is you take at a particular time, the social network, it's a network, right? You can map it to persistent homology, and then you can look at the pairwise distances between the corresponding persistent diagram, and then you can project it back onto, in this case, we're using MDS. So we kind of try to look at like this height here corresponding to the coefficient of MDS, which corresponding to try to capture the differences or the interesting social networks during this email as time goes on. So if you see any points that I look at this mapping is corresponding to part of the day where there is a lot of email traffic, for example. And then there is sort of interesting sort of topological changes in my social network. Versus in the bottom here, those are really during at night, very late at night, where there's not much of activity, so that they have a less of a significance in terms of topological changes in the social network. So the idea is you can using again persistent homology to study time varying data in general. And in this case, you can basically use the persistent homology profile to classify or study structure transitions between social network. There's other ones that, again, this is, again, a picture of tokens embedding, right? So you have different type of signal on top. The middle picture is the tokens embedding, which is a delayed window embedding we have been talking about. And it's, again, a projection of that embedding. And the third one is looking at the persistent diagram of those embedding. So one question is, well, can I just use this persistent diagram for things like classification, right? Because instead of classify the original signal, I'm going to classify the corresponding persistent diagram of the tokens embedding. Okay, so there's also application of this in mechanics, right? So again, this is interesting, is that there is some sort of mechanical process called chatter, and it has to do with how the, I don't know all the mechanical details of it, but it has to do with, think about it as a sort of phase transition. And what they did is that this is a picture of how maximum persistence of the data coming from. So they collected persistent diagram, but they only choose the point with maximum persistence and under different parameter setting in chatter study. And then they kind of look at essentially how the maximum persistence help to give you a decision boundary between chatter and not chatter. Right, so what you see on the left is sort of this sharp separation corresponding to sort of the phase transition between chatter and not chatter. Okay. So which is also quite interesting. The next one is people to use, again, tokens embedding to reconstruct dynamics. So what dynamics are we talking about? The classic on the left, this is what's called Lorentz attractor. So it's a particular very simple a dynamic system, and if you kind of throw a particle in here, it kind of moves around like this. This is how the trajectory moves. And then what you again do is you can sample points from this point cloud. Let's say you have some sample of points from a dynamic system. The question is, can I just reconstruct the dynamic system from the point cloud itself? Okay, this is actually quite non-trivial because in some sense, when I actually just sample points from the point cloud, there's certain information that is lost from the process. So the question, can I reconstruct this? So there's many applications. There's like specifically, this is a particular type of complexes that we actually have not talked about. We have talked about Rips complex. We have talked about Chex complex. right? And later on, we are going to talk about Delaunay complex when we talk about protein docking. But those are just different complexes, simplicial complexes that connect points by edges, triangles, tetrahedra, and so on. But witness complex is a special interesting scenario where what you get is that if I have a point cloud, I am going to choose some form of anchor points and using the distance to those anchor points to construct my filtration. So that's really the high level picture. But what I showed here is actually when I am kind of having more and more points sampled from my Lorentz attractor, how does my witness complex looks like? So of course, as I increase my sample density, I will eventually get, in some sense, a much better reconstruction. Okay. So there's a lot of interesting question for sort of TDA and time series analysis. Mostly I focus today is on the application, but there's also a lot of algorithm and theoretical question. While the obvious question is when I'm doing my tokens embedding, for example, in here, what is my parameter choice? How do I choose the right window size and right gap size? So there has been still active research in that direction. And of course, for a very simple signal, like say, for example, the signal coming from something looks like a cosine sine curve, there has been some good understanding of relating the periodicity of the signal with the right window size. So there, and with actually the persistence of the final signal. So there's a bit of a theoretical advancement in that direction, but I still think there's a lot of potential applications of using topology tools in the time series analysis domain. Especially you want to combine topological tool with maybe the classic signal processing tools. I think that combination can be quite powerful. Okay, all right, any questions? Okay, so if there's no question today, we are going to move on to one more application of TDA. This is part of my TDA plus X series. The next lecture, we're going to talk about topological tools used for protein docking. Okay. Yeah. So, okay. All right, I'm going to stop.